Pharmacovigilance is currently facing a mathematical breaking point. The system designed to monitor drug safety is being overwhelmed by an avalanche of raw medical data. The foundation of this system is the Individual Case Safety Report. An Individual Case Safety Report, or ICSR, is a structured record documenting a single patient experiencing an adverse reaction after taking a medication. These reports are the frontline defense for identifying post-approval drug risks. Historically, processing these reports relied entirely on human labor. Medical assessors had to manually read, triage, and medically code every single narrative that entered the system. That manual baseline was built for a different era. Today, global repositories like Vigibase, the World Health Organization's global database of adverse event reports, and FAERS, the FDA's adverse event reporting system, are taking in data at a rate that exceeds linear processing models. This chart illustrates the volume of adverse event reports entering Vigibase over 2 decades. What used to be a steady, linear climb has accelerated into a steep exponential curve. Overlaying the FDA's FAERS database reveals identical geometry, a systemic, global volume crisis. Scaling human headcount to match an exponential curve is a mathematical impossibility. Relying on manual assessment at this volume causes a degradation in both the speed and the quality of medical review. Integrating artificial intelligence provides the structural capacity to handle exponential data growth, ensuring critical safety signals remain identifiable within unprocessable noise. This flowchart maps the legacy signal management process. The bottleneck begins at the first node, the intake phase. Here, inbound safety data arrives as unstructured narrative text, ranging from dense clinical notes to fragmented social media posts. The core engineering challenge is transforming this unpredictable syntax into clean, machine-readable taxonomies. 2 of the most critical taxonomies are MedDRA, used to standardize symptom descriptions, and ATC codes, which classify the active ingredients in medications. To solve this, natural language processing models act as the first layer of the upgraded pipeline. Natural language processing, or NLP, is a branch of artificial intelligence that allows computers to understand, interpret, and organize human text. These NLP models parse complex medical jargon. They automatically extract patient entities, event narratives, and medical histories. Deployed AI systems execute this automated coding with high accuracy, translating chaotic words into clean, structured MedDRA codes. By automating the structuring of raw data, the primary processing delay is eliminated. Pharmacovigilance scientists can analyze the data immediately, rather than spending weeks formatting it. Once the data is structured, it exposes a critical vulnerability in global databases: duplicate reports. A single adverse event is often submitted separately by the patient, the attending physician, and the manufacturer. Visually, this creates a deceptive cluster. A single reality duplicates itself within the dataset, forming a false anomaly that skews the data. Legacy rule-based duplicate detection simply fails here. Simple matching rules cannot account for slight variations in spelling, differing date formats, or varied narrative descriptions of the exact same event. The technical solution is probabilistic record linkage. This is a machine learning technique that identifies distinct records belonging to the same entity across different data sources, even without a shared unique identifier. This algorithm weighs the mathematical probability that 2 disparate text narratives actually describe the exact same real-world patient. It evaluates fuzzy variables like age and dosage dates, calculates a probability score, and merges them. This specific pre-processing step is absolutely non-negotiable. Dirty, duplicated data will compromise the advanced signal detection algorithms operating further down the line.
Machine learning deduplication acts as a vital filtration system. When the pipeline flags a safety signal, analysts know it is based on unique, verified patient realities, not administrative echoes. We move deeper into the pipeline to its core function, signal detection. This is where the system attempts to find actual adverse drug reactions hidden within millions of clean, deduplicated reports. The legacy approach to this step was statistical disproportionality. This mathematical method flags an alert only when a specific drug event combination occurs more frequently in a database than its historical baseline. The central flaw of simple disproportionality is its inability to account for complex, multivariable confounders. It struggles with geographic anomalies, concurrent medications, and hidden drug-drug interactions. This visualization shows the modern upgrade, unsupervised machine learning. It maps the data into a multidimensional rotating network graph. Clustering algorithms group disparate, seemingly unrelated adverse event reports, mapping hidden correlations that human analysts could never spot manually. Predictive models then evaluate these newly formed clusters, calculating a specific causal association likelihood for each potential safety signal. The algorithms prioritize the outputs, pushing only the most mathematically threatening signals to the top of the queue for immediate human review. Unsupervised learning provides the multidimensional visibility required to identify subtle multi-drug interactions that exceed the limits of traditional reporting ratios. The bleeding edge of this pipeline involves generative AI and large language models. An LLM is a deep learning algorithm trained on massive datasets to recognize, summarize, and generate text. In pharmacovigilance, they execute zero-shot summarization of massive complex safety dossiers, but this introduces the black box problem. The internal decision pathways of a deep learning matrix are unreadable to the humans operating it. This creates a central tension. LLMs are inherently opaque, but global drug safety regulations legally mandate absolute auditability and transparent decision-making. To deploy these systems legally, the industry relies on explicit regulatory guardrails and guidelines developed by global health authorities like the FDA and the EMA. This 2x2 model risk matrix, detailed by CIOMS, dictates the regulatory oversight. It plots model influence against decision consequence. A standalone model making decisions with high patient consequences falls into the extreme high-risk quadrant. Models in this quadrant require strict architectural mitigation, specifically a human-in-the-loop workflow. Human-in-the-loop is a system architecture where a human operator must review and approve an algorithmic output before a final decision is executed. In production, the generative AI system synthesizes the data and generates queries, but a human medical assessor reviews the logic and makes the final causality determination. Algorithmic transparency and human oversight are not bureaucratic hurdles. They are the foundational engineering requirements necessary to legally deploy high-impact AI. This flowchart maps the completely re-engineered pharmacovigilance pipeline. Exponential data noise enters the system, is structured by NLP, purified by machine learning deduplication, mathematically surfaced by unsupervised clustering algorithms, and delivered to a human assessor for review. This new architecture replaces a slow, linear data entry operation with an immediate multidimensional analysis pipeline. The goal is precision intervention, identifying a distinct threatening signal and isolating it before it has the opportunity to scale. This risk-based architecture allows pharmacovigilance to operate as a predictive safety engine, intercepting adverse reactions while they are still isolated data points.